Good morning, everyone. Thank you for joining us today. So this webinar is titled Unlocking Genomic Frontiers in East and West Africa. And so we are just going to be introducing the partnership between Oxford Nanopore and the Lasec Group. My name is Delana Turon. I am the Next Generation Sequencing Application Specialist at the Lasec Group, responsible for Oxford Nanopore Technologies. And so in this webinar, we will be discussing how Oxford Nanopore Technologies is bridging the gap in sequencing technologies in Africa by paving a way for groundbreaking research on health and healthcare advancements, and how the Lasec Group and Oxford Nanopore collaboration is going to empower African scientists and researchers. And with, of course, with Osman Tariq, the sales director from the MIAE region, he will speak shortly. If you have any questions, please put it in the chat while we present, and we can address them at the end of the talk. So just to give a bit of background on Oxford Nanopore and our relationship. So essentially, we partnered with Oxford Nanopore in September of 2023. So quite a new partnership, but it's a partnership that we are very excited about. And so as you can see on the map here, we have territories from West and East Africa, and we cover quite a large majority of it. So just to give a bit of background on Lasec as an established partner, we are based in Cape Town, South Africa, and we were established in 1945. However, you can see in 1990, we did one of our first exports into Africa. And so we've been in this industry in Africa for over 30 years. And so we really understand the market extremely well. So just to highlight what I mentioned on the previous slide, our focus is really on the rest of Africa. We have dedicated sales teams which travel into country constantly. We have dedicated specific country managers, as well as product and technical specialists as myself that to provide you that after sales support, which we really pride ourselves on. And with this, we also have additional customer support with product registrations, in-country documentation, and so forth. And with that, of course, dedicated in-house logistics with freight forwarding specialists. And so just to give you a bit of background on the sales force of the individuals who actually will be working with individuals in East and West Africa. We have about 21 staff members dedicated just for these regions, and that consists of internal and external account managers, which are country specific, as I mentioned before. We also have a range of product managers which address all our portfolios, ranging from consumables to instruments, all the way from biotechnology. And for ONT specifically, we have an application specialist, myself, which will give you that technical support you need after sale. And so we are aimed at providing winning technology and science to all Africans. And so if you have any inquiries, please email us. We can have a call and discuss your needs at ont@lasec.com. So now I would like to introduce you to Osman. Thank you.
Hi, Lana. Thank you for the introduction. Yeah, wanted to say thank you to everyone for joining the call today. I think it's a great opportunity for us to really highlight our new partnership with Lasec. We see Lasec as a key partner for us in expanding our portfolio and presence within Africa. Africa is a key market for us, which we've been looking to penetrate over the last couple of years. The IPO that Oxford Nanopore underwent a couple of years ago has really helped accelerate that as well. So we're hoping that with the new partnership, we're able to grow our presence and help aid the research that's happening in Africa right now. So I'll just start off by pulling up my presentation. So Lana, now I can't, it doesn't look like I can access my presentation now. When we tried before, it was okay, but it seems as if it's gone.
You have that share screen option.
Yeah, but it doesn't let me pull up anything. Let me try again.
Just make you a host.
Yeah, that might be what's missing.
Okay, try now.
Give me a sec. Let me just.
Osman, alternatively, you can send it to me and on OneDrive, and I can share it from my screen and just slides.
Oh, I think it's working now. Yeah, that's all right. Thanks, Rochelle. Looks like we're all good. So let me just share the screen. Let me know when you can see it, please, Lana. Okay, great. So yeah, in terms of the presentation today, it's going to be very much a top-level view of our technology and how that's going to be hopefully aiding the African market. Agenda-wise, a quick overview of Oxford Nanopore itself, a little bit about our technology, and then really the focus area is going to be around the benefits that nanopore sequencing brings to end users and the research community. In terms of questions, if there's any questions, please put them in the chat box, or you can email the ONT inbox that Alana shared with you as well, and we'll be able to respond via email. So in terms of Oxford Nanopore, the organization was set up in 2005 as a spin-off of the Oxford University. We're still headquartered in Oxford, and actually our manufacturing, I think it's really important to highlight that, still happens in the UK, in the Greater Oxford area. In terms of quality control as well, all our flow cells are manufactured and also quality controlled out of the facility there as well. So I think that definitely helps add a USP to our technology. In terms of, if you have had, any of you have had the opportunity to listen to some of our talks, we regularly talk about being a disruptor in the sequencing market. We want to be able to decentralize and democratize sequencing for anyone, anywhere, anytime, basically, which means that lower income countries or more developing economies have the ability to have access to sequencing platforms. If you look at our key focus areas in terms of market coverage, research is definitely an area that we are strong in when it comes to specific things like microbiology, human genetics, and cancer research. We also are involved in more industry-related areas like water testing, forensics, for example, as well as biopharma. Education is an interesting one. We are actually, well, we have a specific department set up to support secondary and tertiary education programs, looking at bringing sequencing into those specific areas. So we work on what I would call government initiatives as well. It's something we're looking to bring into Africa. We do quite a bit of that now in the Middle East, which also falls under my remit, but that isn't a focus area for us. Healthcare and clinical is definitely an area we're looking to break into when it comes to things like infectious disease, oncology, and also reproductive health. Population genomics, we have some large-scale programs happening in the Middle East with a company called G42. I'm not sure if anyone has been following the developments there, but also in the US and Europe. We are working on a couple of projects in Africa. Now we're hopefully going to sign off on the Egypt population genomics project in the next couple of months. So it's definitely an area we're looking at expanding into in Africa. I know Rochelle, my colleague who's based in South Africa, is working with the Tunisia and the Algeria population genomics team as well. So it's an area we're looking to really accelerate into. So when it comes to our technology, I'm sure most of you know, we look at long read technology. The areas we look at really are whole WGS, whole genome sequencing, whole transcriptome sequencing, targeted sequencing, and metagenomics. The long read really allows us to look at areas like structural variants, methylation, gene expression. I won't go through all of these, but these are just highlighting some of the key investigation areas, SNVs, phasing, splice variation as well as single cell. This is a really interesting slide, and it's always nice to show this to our customers to put things into reality, as it were. So our smallest device, which I'll come on to a little bit later, which is the MinION. You'll see that it actually has applications across different areas, but the portability of it, and I think that's important to highlight, areas like Africa and the Middle East, we see a lot of our sales coming through the MinION range. You actually see that it's based, well, you can find our technology in different areas. So you'll see in the bottom left corner here, hopefully, you'll see our MinION, which is based up at the International Space Station. We also have our MinIONs undersea in the ocean, but also at the Arctic as well. So find it in multiple locations, anywhere where things are portable. That's where the MinION really fits nicely. In terms of our collaborations and our customer base, we're very much a global company. And as I alluded to earlier, I think the IPO has helped us really drive our ability to scale up on a global level. So If we're looking at the UK specifically, which is our home territory, we work very closely with people like Public Health, Oxford University, Cambridge, UCL. In the US, we have a strong foothold now as well with Johns Hopkins, Stanford University, for example, NASA, the CDC. On a more local level, I think, Africa CDC is a big one for us as well. So our technology is everywhere. We just need to make sure, obviously, in Africa, with the help of Lasec, we're able to accelerate our growth. And I mentioned G42 as well. They're our biggest population genomic sequencing service on a global level. So in terms of publications, I think it's this graph here specifically, it's a nice one to highlight. So up until 2014, the number of publications we had was very limited. You'll see from about 2019, post-COVID, or when COVID had started to happen, we saw an exponential growth in our publications, which shows that actually our technology is being adopted a lot more. So up until the back end of last year, we had just under 2,400 publications, which on average means we're seeing 38 new publications a week. This graph is really interesting as well. So if we look at 2016, majority of our business, if we broke it down into a segment split, sat with microbiology. So 60% of our revenue was coming basically from, or our coverage was coming from microbiology. If we look at where it was at the back end of last year, that reliance on microbiology has dropped, and we've now shifted more into environmental, animal, plant, and also human genetics, and that will continue to be the trend. So just a quick snapshot of ONT. So our global operations, as I mentioned, basically happen out of Oxford. Our manufacturing all happens in the UK. We're now starting to have a stronger presence in the US with offices across New York, San Francisco. Asia is also a big area for us now, specifically Asia Pac. So we have Shanghai, Beijing, Tokyo, Singapore. We also have, and it's not on the slide, but we set up an office a couple of years ago in Abu Dhabi as part of the G42 collaboration. We're just about to open a customer service center and a sales office in Dubai. I think that's going to be really exciting for our expansion plans. India also, which falls under my remit, we are just about to open an entity there as well. So the point being that our global presence is going to increase, and Africa is an area we're looking to expand into, specifically through our distributor network, and Lasec obviously falls into that. If we look at our technology, 4 main platforms, I'm only going to really focus on 3 today, which are applicable, which is a MinION range, the GridION range, and PromethION, and I'll come on to that in subsequent slides. I think it's important to just highlight once again, our sequencers are now very much present on a global level. So we have over 100 countries where you'll find our sequencers, and also we have strong collaborations and initiatives with key global institutes. Application-wise, I won't go too much into it. I've already touched on this, but over 2,400 applications to date, a broad number of applications that can be carried out using our platforms, anything ranging from population genomics, infectious disease, ag bio, environmental, education. We've always been really focused around scientific research, but we're also looking at moving into more applied markets. I think also, a real area where we started to focus and accelerate our growth was the approach we took to sequencing when it came to COVID. That really helped give us a platform to launch our or increase our exposure around the globe to our sequences. In terms of our committed continual innovation and excellence is something we pride ourselves on. We have over 1,300 applications, most of which, half of which have been developed in-house. We continue to partner with global academic partnerships and also drive innovation through supplier relationships. So in terms of our technology, and like I said, I won't go into too much detail, but fundamentally our technology is focused around our flow cells. So it's actually not the platform itself, it's the flow cells where all the magic happens, as it were. So this is a protein nanopore. This is what is patented by Oxford Nanopore, and you'll see that actually this is where you get the DNA or RNA molecule going through the nanopore, and that's where all the sequencing happens. This picture gives you a better understanding. So you have the nanopore here, which sits on basically the support membrane, and as the RNA and DNA passes through, there's an electrical impulse, which once disrupted, then starts to read that signal and translate that into a specific DNA and RNA sequence. And this is all real time. So as soon as you basically load the sample and the sequencing starts, it all starts to basically pull that data live. So why nanopore sequencing? So there's 6 really points that I wanted to highlight and takeaway points really for the audience. The first one is direct sequencing of DNA and RNA. So we are able to sequence native DNA and RNA, which means that actually we're removing, there's no real requirement for a PCR or amplification. You can do that if you want, but with anything when it comes to amplification, for example, you're losing quite a bit. You're introducing a bias, but you're also missing certain modifications as well. So with our sequencing, native sequencing of DNA and RNA, that is, that's excluded from that situation. Also, we're not only a long read company. I think that's a misconception sometimes. Our competitors like to bring up, which is that we can't cover short read. That's wrong. We cover the short read and long read, so we're able to pick up things, as I mentioned earlier, like genomic variations, repeat regions, phasing, methylation as well. The longest read is 4MB. Real-time analysis, I think that's really, really important as well. So as soon as you start to sequence the DNA and RNA, you're able to pull up data as the unit or our platform is running. You can start, stop as well, which I think is really important once you've got what you need. And also an area we're looking into now and we're starting to work more on is adaptive sampling, which I think will definitely help the research community. Scalability, I think this is really important when it comes to places like Africa as well. So we have multiple platforms, and depending on your budget, depending on your requirement on what you're analyzing, you're able to buy a unit or a platform that is specific to what you need. And also the workflows are transferable. So if you have the MinION, for example, you can scale that up to a GridION, to a PromethION as well. And experiments can be QC optimized on the smaller devices and then scaled up to the larger devices. A flexible library prep, once again, really important as well. I'm sure if you've been using technologies like Illumina, for example, you'll see that the actual library prep takes quite a bit of time. Ours is rapid in that sense, so this definitely helps speed up the process, and actually the ease of the library prep is quite nice. Flexible purchasing options. Listen, we're all constricted by budgets, and areas like Africa where definitely we see that budget is more of an issue. You're able to buy a sequencer for as little as around about $1,000, and it kind of scales up. If you're looking at the PromethION range, it's more around about the $200,000-$300,000. So there is a platform for every researcher and research requirement. We also offer a CapEx and a non-CapEx model. So what we call starter packs, where you don't actually pay for the unit, you just lease that for a couple of years, and that's maintained by making sure that you keep up with your license and warranty on a year-to-year basis, or you can buy CapEx. So there's a lot of flexibility in terms of what we offer. So this is a picture showing you all the platforms that we have. So as I mentioned, anything from a small fungal or a MinION, which is basically USB size sequencer, all the way to our benchtop PromethIONs, which is more used for high throughput work. So this gives you a better understanding as well of the workflow here. So we have the MinION, and under the MinION range, we have the MK1B and the MK1C. If you're looking at maximum output, looking at 50 GB per flow cell for the MinIONs. For the GridION, that goes up to 250 GB. This is all based on a 72-hour run by though as well. So I think it's important to highlight that. And then when you're looking at the larger scale sequences, the PromethION P24 and P48, we do have a P2. Unfortunately, it's not part of this presentation, but we do have that, and they all go up to about 290 GB. So just a little bit more detailed in terms of the specific different platforms. This just gives you a better understanding of what it looks like. So the MinION, very much like a USB. As I mentioned earlier, the actual science happens here, which is a flow cell, which has the nanopores, and you basically load your sample and the sequencer works. You would plug this one specifically into a basic laptop, and you'll be able to analyze the data that comes out of it. The MinION, the difference, MK1C, sorry, the difference between the MK1C and the MK1B is that actually this has an onboard compute. So you're able to see live data being streamed as you're performing the sequencing. We then have the GridION, and the main difference between the GridION and the MinION is actually that here you have 5 positions. So you have 5 different flow cells that can be loaded. The beauty of this is actually you don't need to batch load. So people like Illumina, for example, some of the other competition, you need to batch load to get economies of scale when it comes to using your flow cells. Here, you can actually run 5 different experiments, start and stop at different times as well. So that gives you that extra ability to have flexibility and also save money. This as well has an onboard compute, which I think is really important to highlight as well. So in the back here, we have an NVIDIA basically system, a chip that's loaded. So you're able to actually do the data analysis through the GridION platform. And then we have the PrimiThine, which is really for our high throughput customers. As I mentioned, we have the P2, P24, P48. The 24, 48 relate to actually the positions or the number of flow cells that are applicable. We also have this A100 unit, which is the compute unit. So it's not an integrated unit, but the A100 comes with the actual PrimiThine P24 and P48. The P2 doesn't. That's more of a standard unit, but you can link that up with a GridION if you want to have a compute. Or we're actually going to launch the P2i, which is the P2 integrated next year, around about Q2, which will have the integrated system, compute system as well. Flong, I'm not going to cover too much. It's not really relevant. In terms of accessibility, as I mentioned earlier, we're very flexible in terms of how we support our customers. So you have the option of a CapEx or a starter pack. Starter pack, once again, is where we lease the unit to you. You commit to buying a certain amount of flow cells that you have to basically call off within a 9-month period. So that gives you that flexibility of not having to pay a high level of CapEx. Pricing is very transparent. We have an online website where you can see the pricing. We are working, and I know Alana and the team are excited about this, but we are working on a better pricing structure for our channel partners to help incentivize them to grow their sales. As I mentioned, no bashing required, so there's no penalty for not using every single flow cell that's located in, for example, the PrimiThine. So just to kind of summarize, really simple workflow when it comes to sample preparation for the DNA and RNA. We're able to scale up by going from the Flongle to the MinION to the GridION to the PrimiThine. We also have our own software platform, which is called Epitome as well, which helps real-time analysis. Once again, unique benefits, and I highlighted this earlier, direct or native sequencing of DNA and RNA. We're able to not only do long read, we can do ultra long read, we can also do short read, real-time analysis. You can start and stop as you analyze. The scalability, portability in areas like Africa, definitely really important. So you can start off with a USB held device and scale all the way up to the larger desktop devices. Mentioned the flexibility of a library prep as well, and also more importantly, I think the option that we give customers with CapEx and non-CapEx definitely helps support what we're trying to achieve in Africa. So yeah, that was a quick overview of our technology and the platform. And
thank you, Osman. I want to thank you all for joining, and we hope to speak to you soon. And have a lovely day further. Thank you, Osman.
Thank you, everyone. Take care.
Bye.
Bye.